Okay, so these are some of the mechanisms that our body uses to regulate blood glucose. So we're going to talk about metabolic regulation, and this has to do with both transporters and enzymes. So we're going to look at this. So this kind of starts off with this generic diagram here of a cell. And glucose entering the cell, and then being modified to glucose 6-phosphate. And so if we talk about the liver first, and so let's say this is a liver cell. And remember from that table we said, well, we know that liver cells have GLUT2. We said GLUT2 is not affected by insulin, right? But we said it's concentration, gradient dependent, it's bidirectional. And so why would the liver cells use GLUT2 instead of some of the other transporters? And here's why. GLUT2 has appropriate characteristics that serve its role in liver cells. So I'm going to tell you basically, I'll tell you 4 reasons, or these are 4 characteristics of GLUT2 being appropriate for liver cells. Has a high Km. Does that mean it? Right. Oh, right. High Km. We've all been, right? We've all been. I was in those lectures where I went, yeah, what's a Km? Oh, it's K-less mint. And I'm like, all right, I'm in nutrition. Right? So we'll talk about that in a second. For now, let's just say, high Km means what? High affinity or low? Low. So let's say this now let's put it this way. How about this? And any enzymologists, I apologize, but this is an easy way I think of looking at it. It's high capacity. In other words, it's a fast transporter. High Km is high capacity. So it's a fast transporter. It's bidirectional. Go either way. It is not insulin dependent. Not insulin dependent. I'll put all these together in a second. And number 4 is also transports fructose and galactose. Remember, GLUT2 is on that basolateral membrane of intestinal cells. That's one place for it, and that's great because then it can move all 3 monosaccharides. Same thing is advantageous in the liver cells, right? We want fructose and galactose to go into liver cells. And one of the reasons is because the liver can convert those to glucose. So it also transports fructose and galactose. Right, so you can say, it's kind of a promiscuous transporter. It'll do any of the monosaccharides. So it's good to have on the cell surface, especially here because the portal vein is going to be taking those monosaccharides from the diet, from a mixed diet, and the liver can incorporate any of those by moving it in through GLUT2.
How does the high Km equal low affinity?
So in this case, let's just say this is a protein. You guys probably looked at these equations where you looked at enzyme plus substrate and all that, right? So let's say this is my substrate. Let's just say this is my enzyme. In this case, I'm going to say it's GLUT2. Right, you looked at it when you're talking about this in enzymes. You said, well, if that binding site or that catalytic site for that substrate is a tight fit, then basically it's a closer, tighter interaction with substrate, which means it's very specific, but it binds to it tightly, which means it's slower in releasing it. And you can look at the same thing for transporters, right? In this case, we have a transporter that has a low affinity, so that it can move it through quickly, which means in some cases that's a little less specific. And the evidence for that is, yeah, it also does fructose and galactose. Okay? Now, let's talk about why this is appropriate. So, and I haven't given you this, but you'll see this next quarter. We said the fasting blood glucose concentration is 70 to 110 milligrams. You know, that's if, for example, you do a blood sample from your arm or your finger or something like that, systemic, right? Now, what about after a meal? You eat a meal, let's say it's got a lot of carbohydrate, and now let's say I waited about 15 minutes and then I started seeing my blood glucose go up by doing finger pricks. And I say, wow, it's gone from 80 to 220. That's a significant increase. That's here in my fingertip. Guess where it's a lot higher than that? Portal vein. It's not diluted. That portal vein is taking those monosaccharides directly from the gut and now moving them to the liver. So it's a smaller system. It's not diluted. It's all coming in, and now all that high concentration of glucose is going to the liver. We don't want to overwhelm that transporter. So in that case, we say that high Km allows the liver to absorb a lot of glucose postprandial. Okay? So it's not going to be overwhelmed. What keeps it from pulling out all that glucose? Low affinity. Right? And we'll talk about that a little bit later when we talk about Km curves. Remember that? Km equals the substrate concentration at 1/2 Vmax. Probably think about that every day. Today's a good day because the Km still equals substrate concentration at 1/2 Vmax. I can draw that on right before. Yeah, I draw that. Oh yeah, it goes like there's my Vmax, there's my half my concentration. Remember that? We'll do that maybe next time. And then you go, oh yeah, why does that matter? Because when you have glucose in their portal vein and it's going to the liver, that Km matters. Okay? I'm not going to make any jokes about biochemistry, but, right? So that matters. What about this? These two are kind of together. We already talked about this, right? That's important because now you got the same transporter that can move fructose and galactose, and it's got high capacity. So if you have all of these in there at the same time, these can go in at about 100%. Okay? What about these? Why is it important that it's bidirectional, it's not insulin dependent? Think about when you're taking it in. When you take that blood glucose in, and now essentially you want that liver to be able to take in glucose. You don't want to wait for insulin. And what I didn't tell you is, and I'll tell you right now, is the portal vein is also exposed to the pancreas. So the pancreas, it gets exposed to the concentration of glucose after a meal, that increase, very quickly. Okay? But now if we put these together, now let's say fasting. Let's say we're 6 hours away from a meal. We haven't eaten for 6 hours. We're in kind of a fasted state. The liver will support that blood glucose concentration during fasting by putting glucose into the blood. How does it secrete it into the blood? Through GLUT2. So this allows bidirectional means that the liver can secrete glucose into the blood to support other tissues. Right, during fasting. Sure, during fasting, the GLUT2 allows the liver to secrete glucose into the blood. During fasting, is insulin high or low? It's low. Doesn't matter. Got it? It's not insulin dependent. So during fasting, when insulin is low, GLUT2 is still active. See the way all those work? And again, I would say, I mentioned this before, but try to kind of put it together in a way that makes sense. And our body's is pretty, the corny term is amazing. If you think, oh, it should probably work that way, it probably does.
GLUT5 has a higher affinity for fructose, but if GLUT2 has such a high capacity, why is there GLUT4 in the enterocytes?
Why is there GLUT4?
I mean GLUT4, sorry. GLUT5 in the enterocytes. Oh wait, I'm sorry. I'm looking at SGLT, whatever. I'm tired.
To move, yeah. Okay, so let's see. Okay, so now let's say, for example, it goes inside the cell and it's converted to glucose 6-phosphate, right? Basically, there's an enzyme here that takes a phosphate from ATP, adds it to glucose. This particular enzyme adds it to the hydroxyl group on carbon number 6. So we call it glucose 6-phosphate. Can it go this way? Why can't it go out? Now it's GLUT 6. It's glucose 6. It's no longer glucose. Glucose 6-phosphate is not substrate for GLUT2. So now think about what the liver can do. It can take in glucose. This is a bidirectional transporter. If it doesn't modify it, that glucose can go this way. So the liver traps it. Remember that term metabolic trapping? That's what we're talking about here. That's when a molecule is metabolized to a form that it traps it. And that's all the liver does. It goes glucose 6. Now I can metabolize this if it's not going to be released. And so we'll talk about all that coming up.
So does it then convert it back to glucose?
It can. Liver can do that. It's probably not going to do it in that case. We'll talk about why, but it can. Like it can go all the way from pyruvate, all the way reverse glycolysis, get to glucose 6, and the liver can convert it to glucose and kick it out. Muscle cells won't do that. So if a muscle cell does this, it doesn't come back out. And I'll point that out coming up. So let's talk about this. Phosphorylation of glucose is also a regulated event. Remember that term kinase? What's a kinase? It's an enzyme that adds phosphates to other molecules. That category of enzymes. So kinases add phosphates to other molecules, so we say it's phosphorylated, right? And there's a lot of these kinases. We're going to talk about 2 of them, hexokinase and glucokinase. They're isoenzymes. In other words, they do the same reaction, but they're expressed in different tissues. So let's look at glucokinase and hexokinase. If it does the same reaction, why wouldn't you just have one enzyme? Because both of those different forms of the enzyme have different characteristics. So I'm just going to go like this. All right, here's ATP, ADP. So So when you draw this for me, don't forget your ATP. Glucokinase and hexokinase. Glucokinase is expressed in the liver and the pancreas. Glucokinase is expressed in the liver and in the pancreas. Glucokinase has a high Km. Glucokinase has a high Km. All right, so we know that's a low affinity. Hexokinase. I think we can say extrahepatic tissue. What does that mean? Extrahepatic tissue. Everywhere except the liver and pancreas. Pretty much everywhere else. So far, glucokinase is that. Hexokinase, low Km. Right, that means high affinity. And what else? It's down-regulated by glucose 6-phosphate. What do we call that? Feedback, negative feedback, feedback inhibition. All right, it's basically slowed down by product.
So you said both of them are in the liver and pancreas?
No, glucokinase is in the liver and pancreas.
Where is extrahepatic tissue?
Hexokinase outside the liver. So you could say everywhere except liver and pancreas. Again, why would the body do that? We'll talk about that.
Pancreas is not considered hepatic tissue.
Correct. It's not the liver.
Everywhere.
Yeah. Now let's kind of think about why that's the case. Again, what has happened is the tissues have paired, transported with enzyme. GLUT2, this is a liver cell. Let's say this is a liver cell. This is GLUT2. It's got a high Km. GLUT2 can allow a lot of glucose to come in. And because glucokinase has a high Km, a lot of it can be phosphorylated. In muscle tissue, you're going to have GLUT4. GLUT4 is a low Km. Hexokinase has a low Km. Which means GLUT4 and hexokinase work efficiently at low glucose concentrations. And we'll have fun with the Km curve next time. But GLUT4 and hexokinase work efficiently at low glucose concentrations. Right, and you can just kind of think big picture and go, well, you know what? Even after a meal, the liver's exposed to higher amounts of carbohydrate than muscle. And now think about during fasting scenarios, right? The liver can make glucose. It can export it into the blood. Under those conditions, Hexokinase is able to phosphorylate any glucose it gets in that muscle cell. It works well at a low concentration. It has a low Km, it has a high affinity. Any glucose comes into that muscle cell, it's going to tend to be phosphorylated effectively in muscle. And now you can imagine like the scenario of fight or flight. If you do need to do high intensity exercise and that glucose is in the blood and it goes into the muscle cell, then it's going to be phosphorylated and it can run through glycolysis. Okay, so it kind of tells you about the role of those tissues in the body and why these transporters and enzymes are paired to support the important functions of each of those tissues. Okay. Here's another way that blood glucose is kind of regulated. It's called the Cori cycle. Have you heard of this one? These are actually, maybe in biochemistry, that sometimes they mention it. There's two biochemists that figured this out. It's actually a married biochemical researcher couple, and they figured this Cori cycle out. This cycle provides a metabolic connection between the liver and other tissues that depend on glucose for energy. The examples of tissues involved include red blood cells and muscle tissue and the liver. So if we look at this, here's an example of the Cori cycle, the diagram showing it. So left side is the liver, and the liver can do things that other tissues can't, including make glucose. So here's a red blood cell. In your notes, put or muscle cell. So here's the blood or plasma. Plasma is just the part of blood without red blood cells, right? Glucose enters into the red blood cell or muscle cell, goes through glycolysis. That cell gets ATP out of it. Red blood cells will make lactate. They don't have mitochondria. They can't go pyruvate to acetyl-CoA. So they convert it to lactate and purge it. Lactate runs down its concentration gradient out of the cell. When would that happen in a muscle cell? Anaerobic metabolism, high intensity exercise, you make a lot of lactate. What does the muscle do? Purges the lactate to maintain a pH, because that's an acid. Puts it into the blood, and then other tissues pick it up. Those other tissues include the liver. Now the lactate goes into the liver, and look what it does. Lactate to pyruvate, gluconeogenesis to glucose. This is gluconeogenesis, right? Requires energy, but look what it's doing. It's providing glucose back to other tissues. And now the glucose can basically repeat that. So if you were tracking carbons, some of those carbons would do that over and over again. This is the reason one of the reasons I'm telling you this is it's estimated that approximately 40% of our blood glucose is involved in a Cori cycle. It's 24/7. It's estimated approximately 40% of our blood glucose is involved in the Cori cycle.
Can you explain again why the product of glycolysis is lactate?
If it's a red blood cell, it will go glucose to pyruvate, and then red blood cells don't have mitochondria, and the enzyme that converts pyruvate to acetyl-CoA is in the mitochondria. Yeah, so that's their only game in town. Got to go lactate. So this is a way that tissues are cooperating in glucose metabolism. So if you think about what's happening is, the liver is supporting other tissues by converting lactate back to glucose. I honestly don't know what the liver gets out of it. Because that's kind of, you've heard of that, like the carbohydrate paradox. There's tissues in our body that require glucose but can't make it, can't store it. So the liver is crucial for stuff like this. So here's what it's not. It's not increasing blood glucose. This is not a way to increase blood glucose. Can you see why? You guys see that? Why not? Why isn't there an increase? Yeah, it's just cycling through. So don't say, if I say, tell me how the liver can increase blood glucose. You could say hepatic glycogenolysis, hepatic gluconeogenesis. Don't say the Cori cycle. It's not doing that. It's helping maintain it. It's not adding to it. Right? But I mean, honestly, it would completely change our physiology if the liver couldn't do that. Because red blood cells have to have glucose. All they can do is make lactate from it. And so that's one of the tissues, one of the reasons why we have to have carbohydrate or red blood cells. Uh-huh.
2 ATP that the red blood cell is getting, is that going, that's staying in the red blood cell?
In the red blood cell, yes. Yeah. Uh-huh.
So when a person is in ketosis, how does, or what happens to this cycle?
That cycle has to continue. Absolutely has to continue. Now, so this involves gluconeogenesis. So as we decrease dietary carbohydrate, hepatic gluconeogenesis goes up. Right? And we'll talk about why, but one of the reasons is hepatic gluconeogenesis is regulated by hormones, like glucagon. So when blood glucose starts dropping, glucagon goes up and stimulates hepatic gluconeogenesis. Well, so here's the tricky part. Our brain and central nervous system needs approximately 125 to 150 grams of glucose a day. The liver can only make 125 to 150 grams a day. That's unsustainable. That's why our body switches over to ketones. Because the brain and central nervous system can use ketones while the red blood cells rely on the glucose.
It's not upregulating, it's just like more is being made. That's what you're saying.
Yeah, there's a ceiling for hepatic gluconeogenesis, yeah. And the other thing to take away from this is hepatic gluconeogenesis occurs 24/7. There's always some going on, but at some points it's higher than others. But it's a very important way of regulating glucose. And that's why, for example, some type 2 diabetics are on anti-hepatic gluconeogenic drugs to help maintain a lower blood glucose level. So it's significant, and it can be regulated by hormones and some medications. But Cori Cycle is involved in that. So those are kind of metabolic regulators of blood glucose concentration. Let's talk about hormones. There's quite a few of them that are involved in this. We're just going to look at 3 of them, but mainly the first 2, insulin and glucagon. We know insulin decreases blood glucose, and glucagon increases it. So insulin decreases blood glucose, glucagon increases it. What does epinephrine do? Increases it, right? Epinephrine is released during stress, mostly physical activity. As soon as a person starts exercising, epinephrine goes up. It basically provides fuel to support the physical activity. What's another name for epinephrine? Adrenaline. Where's it made? Adrenal glands. Yep. Norepinephrine's a little bit different. We're talking about epinephrine. Okay? So let's talk about insulin and glucagon, where they're made. Did we look at this before? I don't think I have this in digestive. So this is a classic drawing of parts of the pancreas. And this is, if you just look at, in the pancreas, I said, 2% of pancreatic cell volume, endocrine function. That's mostly beta cells and alpha cells. The beta cells secrete insulin. The alpha cells secrete glucagon. But the nice thing about the system is they're both exposed to the same blood glucose concentration because alpha cells and beta cells are right next to each other. So this is in the pancreas in a specific structure called the Islets of Langerhans, which are these groupings of endocrine cells. And here's a beta cell that will release insulin when blood glucose goes up. Right next to it, alpha cell will release glucagon when blood glucose drops. So 24/7, those hormones are kind of counterbalancing each other to maintain blood glucose within a certain range. And if you look at some of these experiments, this is probably a good caption to be response to fasting. Be careful what you sign up for. These participants started a fast here, and at the beginning of the fast, they measured insulin, glucagon, and blood glucose. Here's blood glucose. You can see it's 90 mg per deciliter, right in the middle of that range. And you can see glucagon's here, and here's insulin. I'd say this is more than the fast. This is starvation, right? Generally, after about 24 hours, we say it's no longer a fast, it's starvation. And now look what happens. Blood glucose is dropping. No carbohydrate coming in. So what's happening? Blood glucose is dropping, and that's triggering what? Glucagon. Glucagon is released, and what are two ways that glucagon can maintain this? It doesn't drop to zero. That's not a good scenario. So what does this mean? How's the body doing this? Hepatic glycogenolysis. Hepatic gluconeogenesis. Hepatic glycogenolysis, hepatic gluconeogenesis are what are keeping that blood glucose from dropping farther. And now look what happens when glucagon goes up, insulin is coming down. Why? Blood glucose is low. So insulin is stimulated by glucose concentration. If blood glucose is low, insulin is low. And the opposite for glucagon. Okay? As we go through this, I'll kind of give you more details of what happens during starvation. And one of the big issues is everything changes when the liver runs out of glycogen. How long does that take? About a day. So you can put in your notes now. Hepatic glycogen typically will only last 18 to 24 hours of fasting. And then what happens? That's when ketogenesis really picks up to provide an alternative fuel source. So it's a survival mechanism. We can talk about more of the issues of that. Any questions on this? So if it's okay with you, I'm going to stop there. And then next time we'll pick up on this. The quiz. Oh, let me stop this.